Hello everyone. From this lecture, we will learn about the material transport function of cell membrane. First, let's briefly review the structure of cell membrane. It is very thin, elastic structure, only 7.5 to 10 nanometers thick. It serves as a semi-permeable barrier that allows the cell to maintain the composition of the cytoplasm, which is far different from that of the extracellular fluid. This figure gives the approximately concentration of the important electrolytes and other substances in the extracellular fluid and the intracellular fluid. Note that the extracellular fluid contains a large amount of sodium, but only a small amount of potassium. Exactly, the opposite is true of the intracellular fluid. Also, the extracellular fluid contains a large amount of chloride ions, whereas the intracellular fluid contains very little. But the concentrations of the phosphates and the proteins in the intracellular fluid are considerably greater than those in the extracellular fluid. These differences are extremely important to life of the cell. The purpose of this chapter is to explain how the differences are brought about by the transport mechanisms of cell membranes. This figure shows the structure of the cell membrane. Its basic structure is lipid bilayer, which is a thin, double-layered film of the lipid, each layer only one molecule thick, that is continuously over the entire cell surface. Interspersed in this lipid film are large globular protein molecules. The basic lipid bilayer is composed of phospholipid molecules. One end of each phospholipid molecule is soluble in water, that is, it is hydrophilic. The other end is soluble only in fats, that is, it is hydrophobic. The phosphate end of the phospholipid is hydrophilic, and the fatty acid portion is hydrophobic. The lipid bilayer in the middle of the membrane is impermeable to the urea water-soluble substances, such as iron, glucose, and urea. Conversely, fat-soluble substances such as oxygen, carbon dioxide, and alcohol can penetrate this portion of the membrane with ease. The cholesterol molecules in the membrane are also lipid in nature because their steroid nucleus is highly fat-soluble. These molecules, in a sense, are dissolved in a bilayer of the membrane. They mainly help determine the degree of the permeability of the bilayer to water-soluble constituents of the body fluids. Cholesterol controls much of the fluidity of the membrane as well. It constitutes a barrier for the movement of virtually all water molecules and water-soluble substances between the extracellular and the intracellular fluid compartments. This figure shows globular masses floating in a lipid bilayer. These are membrane proteins, most of which are glycoproteins. Two types of proteins occur: integral proteins that protrude all the way through the membrane, and peripheral proteins that are attached only to one surface of the membrane and do not penetrate all the way through. Many of the integral proteins provide structural channel through which water molecules and water-soluble substances, especially ions, can diffuse between the extracellular fluid and intracellular fluids. These protein channels also have selective properties that allow preferential diffusion of some substances over others. Other integral proteins act as carrier proteins for transporting substances that otherwise could not penetrate the lipid barrier. Sometimes these even transport substances in the direction opposite to their natural direction of diffusion, which is called active transport. Still, others act as enzymes. The peripheral proteins function almost entirely as enzymes and receptors. Some are cell-adherent molecules, which anchor cells to their neighbors or to basal liminars. Some glycoproteins function in antibody processes and distinguishing self from non-self. The quantities of the compositions of the extracellular fluid are greatly different from that of intracellular fluid, which are extremely important to the life of the cells and are brought about by the transport mechanism of the cell membrane. The transport across the cell membrane is accomplished mainly by simple diffusions, protein-mediated transport, excytosis, and endocytosis. Simple diffusion means the movement of the molecules in the solution from one region to another as a result of their random motion. Since the cell membrane is a lipid membrane, substances that are soluble in lipids such as oxygen, carbon dioxide, fatty acids can dissolve in the cell membrane and diffuse across it from a solution of a higher concentration to a solution of lower concentration. The amount of substances crossing a membrane in a unit of time is called flux. The net flux always proceeds from the region of higher concentration to that of lower concentration. The magnitude of net flux depends on the concentration difference, temperature, mass of molecule, surface area, lipid solubility of the molecule. Oxygen, carbon dioxide, fatty acids, and steroid hormones are easily soluble in lipid with a greater net flux. All molecules and ions in the body fluids, including water molecules and dissolved substances, are in constant motion, each parcel moving its own separate way. Motion of these particles is that physics called heat. The greater the motion, the higher the temperature, and the motion never ceases at any condition except at absolute zero temperature. Facilitated diffusion uses a transporter to move solute downhill from higher to a lower concentration across a membrane, which belongs to passive transport. The energy source for facilitated diffusion is the transmembrane concentration gradient of the transported substances. Facilitated diffusion via carrier. Because the transport of substances in this way need a specific carrier protein helping it. The carrier binding with the substances on one side of the cell, the compound moving through the cell membrane by configurational change of the carrier protein, the carrier releasing the substances on the other side. Glucose and most of amino acids are mainly transported through the cell membrane via facilitated diffusion. Facilitated diffusion via ion channel. Sodium ion, potassium ion, chloride ion, and calcium ion diffuse across the cell membrane through ion channels, which are integral proteins spanning the lipid bilayer. The integral protein has a hole in the middle providing the channel for ion movement. The diameters of protein channels are very small, preventing larger polar organic molecules from entering the channel. Ion channels show selectively for the type of ion that can pass through them. For example, potassium ion channels allow only potassium ion to pass, and sodium ion channels allow only sodium ions to pass, etc. The membrane potential provides an electrical force that influences the movement of the ions across the membrane. The inside of the cell has a negative charge with respect to the outside. There is an electrical force attracting positive ions into the cell and repelling negative ions. Thus, the direction and magnitude of ion fluxes across the membrane depend on both concentration difference and the electrical difference. These two driving forces are collectively known as the electrochemical gradient, also called electrochemical difference across a membrane. Three factors can alter the channel protein conformation, producing changes in the opening frequency of duration, chemically gated channel, binding of the specific molecules to channel protein, producing a change in the shape of the channel protein, voltage-gated channel. The changes in the membrane potential causing changes in the shape of the channel protein. Mechanically gated channel, physical deforming the membrane affecting the shape of some channel protein. In this section, we learned about passive transport. Passive transport is the membrane transport of substances along concentration and chemical gradients, including simple diffusion and facilitated diffusion. The same point of the two methods of transmembrane material transport is along the concentration gradient and does not consume energy. The difference is that the transported substances are different and facilitate the diffusion, which requires the participation of membrane protein. If the substances is transported from low concentration to high concentration side, how is it carried out? That's what we are going to learn in the next section. That's all for this section. Thank you. Goodbye.